All right. I want to go ahead and reward the folks that are here, and others will likely join. But have the great pleasure of introducing Simon Gray, MD, PhD, who is currently a postdoctoral fellow in the research component of his GI combined basic science track GI fellowship um, studies. It's been a pleasure to work with Simon. He has a absolutely stellar background with accolades at every level, getting his chemical engineering degree at University of Maryland, Baltimore County, then spending a year in Cambridge, England, where he got a master's in a combined program of chemical engineering and kind of business innovation, oriented toward applications in medicine. Then started a combined MD PhD program at the Yale School of Medicine, where he got a PhD in immunobiology with the absolutely stellar immunobiology group at Yale. And then we were fortunate enough to recruit him to Chapel Hill to do a PSTP accelerated program with internal medicine and now gastroenterology, hepatology. And so what I remember from Simon's application is that he is a sailor, volunteer, a beekeeper, and an Eagle Scout. And so that kind of sums up this is an all-American good guy. So anyway, I am delighted to, and he came as advertised, I would say. So it's been a pleasure to host Simon, and I'm thrilled to turn it over to him to have him talk about a component of the research that he's been doing.
Thanks so much for that introduction, Balfour. So I'm excited to talk to everyone today about the work I've been doing over the past 2 years of the T32 in my postdoc in Balfour's lab, working towards developing a model for studying IBD relapse and understanding the mechanisms that underlie that. I'd like to first just acknowledge a lot of people who have really helped and been critical parts of the project and work I've done. Of course, Balfour is an incredible mentor and incredibly supportive and really excitable and excited scientist. Is really a pleasure to work with in the lab. And the Starter Lab's a very, very collaborative place that I feel very privileged to be a member of, and we all team in on experiments. And really everyone in the lab is, it's listed here as critical parts of the work that I'm going to present. Jeremy Herzog has been probably the most involved in the work I've done that I'm going to be showing today. And a lot of this work is in deep collaboration with a graduate student in Anthony Fodor's lab, who's done a lot of the heavy lifting on the computational work. And I'd also like to thank members of my T32 committee for their guidance, and the Notobiotic course, specifically Josh Frost, especially for his flexibility with last minute requests. So my clinical and research interests are, of course, in inflammatory bowel disease, and specifically with my background in immunology, I'm interested in understanding the role of microbes in the host pathogen, host microbe interaction at the mucosal barrier. And inflammatory bowel diseases are really the perfect place to study that. So inflammatory bowel disease are a huge problem. Over 3 million Americans in America suffer with IBD, and it's a massive annual healthcare expenditure of over $7 billion. The two phenotypes are very different, but what I want to stress is that they have overlapping genetics that are subtly distinct. And within the overlapping genetics of UC and Crohn's disease, there's a subset of patients who have very early onset IBD. So these are children that basically developed extremely severe IBD in the first months or years of life. And I point that out because the genetics that these patients typically have is more of a monogenetic Mendelian type disease with deficiency in the IL-10 pathway, whereas patients who have adult onset disease often have less clear genetic risk factors. But the big picture is that inflammatory bowel diseases really have, in my mind, two components. One is a genetically susceptible patient, and the second is, importantly, an inducer of inflammation, which I hope to convince you is the resident bacteria. So I think it's helpful for understanding the work that we're doing to just take a quick look at the past, present, and then what I think is the future of IBD research. So inflammatory bowel diseases in what we can call the pre-biologic era, so prior to the development of anti-TNFs, were really honestly a surgically managed disease. So Crohn's disease in the 60s, 70s, 80s, and 90s, if you look at old natural history papers, 74% of Crohn's disease patients required surgical resections. And of patients with ileocolonic phenotype, which is a large group of the aggressive Crohn's disease patients we see clinically, 92% of these patients needed resections. And these are really, really high percentages of patients. And the medical treatment options were not great. There was steroids. That was a big breakthrough for treatment of fulminative ulcerative colitis, really to prevent death during hospitalization. But looking at in the 1980s, there was basically no statistical correlation between the type or duration of medical therapy and prognosis. And shockingly to me, Crohn's disease really was a potentially fatal diagnosis of patients who required surgery, of which 74% did. There was a 7.3% Crohn's disease-related death rate. This is in the 80s. Of non-operated patients, they still had a 2.5% death rate, and that's over a 14-year follow-up period. So this is a really morbid disease. Ulcerative colitis, not as morbid as Crohn's disease, but certainly 40% of patients with UC required colectomy. Patients with pancolitis, 61% required colectomy. 10% had toxic megacolon as a complication within the first 2 years. So IBD was a really, really difficult to treat problem, and really mainly treated by surgeons. But that changed a lot with the development of the modern era of therapies, which really took off with the anti-TNFs, with infliximab 25 years ago. But what I want to stress here is that although we've made big strides in the development of therapies for ulcerative colitis and Crohn's disease, the medications we have are not great. So what I'm showing here is medication efficacy as a percent above placebo for induction and maintenance remission in UC and Crohn's. Across the board, is pretty much, and these are in the marquee FDA clinical trials, phase 3 trials. Across the board, there's, the best drug we have essentially, I think, is infliximab. Maybe 30% of patients have above placebo have induction or remission. And that's about as good as it gets. I mean, that's not great. If you had a patient in clinic and you wanted to say, look, this best drug we have only 30% better than placebo at getting you into remission, that's a difficult conversation to have. One of the things I really learned going through fellowship was that while we might be able to, cycling through drugs, get folks into remission, maintaining remission is actually incredibly difficult. So if you look at patients who achieved remission, those who maintain remission, which are in the red, these are ones who were able to induce remission, we're talking low double-digit percentages, less than 20%, pretty much across the board. So big picture here, our drugs have really made a big difference in quality of life and treatment of this disease, and has likely decreased morbidity, but they don't last, and there's high percentages of relapse, and they're not as good as what one would hope. The other issue is that biologics have not clearly altered the disease history of inflammatory bowel diseases. Now, there certainly is evidence that they have reduced need for surgeries. in ulcerative colitis, which I'll show in a second. But if you look at the natural history of Crohn's disease in the biologic era, young children with early onset Crohn's, so this is typically a more aggressive disease, those who had exposure to biologics in the first quarter of their diagnosis, they had a modification in the behavior of the disease, but they had no change in surgery-free survival over an extended follow-up period. In ulcerative colitis, you can see here a 67 to 83% relapse rate in the modern era with biologics. Incredibly high percentage of patients are not maintaining remission with our first-in-class drugs. And although there have been strides made with the biologics, the colectomy rate for patients is still 10 to 20%. So someone walking into clinic with UC, you're going to tell them there's an almost 1 in 5 chance you're going to have your colon removed in your lifetime. So these are really not great disease modifications, and there's really a lot of work to be done. And when we look at towards the future of where IBD research is going, I think that we have to look beyond the single therapy biologics, recognizing we've got a lot of drugs that target different mechanisms, but we have not really seen a convincing disease modification yet in terms of the natural history of the disease. So we really do have to start looking beyond what we've achieved so far. And in my conception, I think there's 4 areas where there's promising opportunities. One is in the diet. This is really an area that clinically we have not at all scratched the surface of, trying to modulate the diet to an anti-inflammatory basis. The second is multiple mechanism combination biologics. Big barrier here is that pharmaceutical companies don't want to fund trials using each other's drugs, but in reality, there's probably benefits to having multiple drugs on board targeting different pathways. So we don't really have any clinical evidence, clinical trials for that. Genetics-guided tailored therapy is likely to be an important way for us to guide patients to different biologics or different modes of action. And what I'm really interested in is microbiome therapy, and specifically live biotherapeutics, which is what I'm going to spend some time talking about today. So this is a conceptualization of what I'm trying to accomplish in my early career development through the T32 and then hopefully into early faculty. What I've tried to do during my T32 time is develop a platform from which I can establish an early career, independent investigative career. And that's what I'm going to be talking about today. This is development of an IBD humanized colitis model to study this phenomenon of relapse in a reductionist animal model. So what I'll be discussing today is the work we've done developing a reproducible humanized model and early work that we've done in developing a proof of concept relapse model, and future work we're going to do in upcoming experiments. And what I hope to do is apply these 2 platforms to really try to understand what the mechanisms and triggers of relapse are, leveraging my background in immunology to understand factors that are really poorly understood, including the antigen specificity. What is it that the T cells are reacting to, clonality of responses, as well as how the microbiome changes in relapse and remission. So getting into the science part of the talk, resident bacteria are required for colon inflammation, and this was discovered in the 90s through experiments using gene deletion mice that developed spontaneous colitis and rederiving them in germ-free settings. So the best example is the IL-10 knockout mouse, which is the work that Balfour did in the mid-90s. But if you derive mice that are genetically normal or deficient in IL-10 and anti-inflammatory cytokine and breed them for their whole lives in germ-free isolators, they have no inflammation. In this germ-free steady state, they have no evidence of colitis. If you were to then colonize them with a complex microbiome, the wild-type animals will not develop inflammation. They develop a robust mucosal immune response, but it's a normal response where the immune system has an appropriate interaction with the microbes, keeps them separate from the body, but doesn't induce severe inflammation. This state of homeostasis is defined by physiologically elevated levels of regulatory T cells that secrete IL-10 and maintain homeostasis. In mice that are deficient in IL-10, which is an anti-inflammatory cytokine, exposing these animals to microbes induces severe progressive colitis. And that's defined in the IL-10 model by interferon gamma in the Th1 helper response and IL-17, IL-23 in the Th17 response. So what this really demonstrates is that microbes are a key inducer of inflammation. But what we know from extensive work by the Sartor lab and many others is that not all bacteria are equal. And this is really important because there are some bacteria that cause very aggressive disease, and they have virulence properties that cause them to invade the epithelium, to secrete toxic molecules, to trigger a very exuberant immune response that's deleterious to the host's mucosal surfaces. But then in contrast, there are very healthy bacteria. So there are bacteria that secrete or produce anti-inflammatory metabolites like short-chain fatty acids that induce regulatory T cells to tone down and quiet the immune response. So these differences are critical for understanding what cause the induction of inflammation in IBD patients. So one of the key ways that we study inflammatory bowel disease is by transplanting germ-free mice with complex microbiomes. And because we know that the microbiomes of healthy people and IBD patients are different, what we do is we transplant feces from IBD patients to mice to study the colitis that they develop and try to understand what it is about IBD patient microbiomes that induce inflammation. So this is the gold standard way to demonstrate a connection between the microbiome and a disease phenotype, this fecal transplant, which we call human microbiome associated mice. But there are limitations to this model or this approach. One is that human sample availability is limited. And so because human microbiomes are highly divergent between people and over time within the same person, the stool that is captured and used in experiments is really a snapshot in time. And what that can lead to is a lot of inter-experiment variability within the same group and between groups at different institutions. And another major problem is that we're trying to engraft microbes and microbiomes that are adapted to the human host environment into mice. And mice are quite different from humans. So there is relatively poor transfer efficiency of the microbes in the human stool to mice. So what we wanted to do was ask the question, is there a way for us to improve this model system and create a more reproducible system by adapting the human microbiome to the mouse environment? So would a mouse adapted human microbiome achieve a higher transfer efficiency and better reproducibility between experiments? So we designed an experiment where we took the feces of 3 patients with active IBD, pooled them and transplanted them to germ-free, genetically normal wild-type mice or germ-free IL-10 knockout mice, which developed colitis in the exposure to complex microbiomes. After a period of 2 weeks, allowing the microbiomes to settle down into a stable composition, we collected daily fecal matter from both these colonized wild type and IL-10 deficient mice and used those feces that we collected over a week to create a what we call a mouse adapted microbiome slurry. And for the purpose of nomenclature, the microbiome that was adapted in a wild type animal, we call a non-inflamed mouse adapted microbiome. And that adapted in an IL-10 knockout animal, we call an inflamed mouse adapted microbiome. So we took those mouse adapted microbiomes and then transplanted them to fresh, germ-free, wild type, and IL-10 deficient mice. and repeated this process 2 more times, serially transplanting the mouse-adapted microbiome across multiple generations. And we wanted to ask, does adaptation of a human microbiome to the mouse environment change the nature of the colitis that mice get? And is that in any way related to the composition of the microbiome? So if you take a look at the colons of mice histologically, you can score the amount of inflammation they have. So let me walk you through what this chart is showing, because there'll be a number of them. So this plot is showing the histologic score that we see under the microscope for different sections of the colon. So we look at 5 different sections, the terminal ileum, and then 4 sections of the colon. And those sections are scored from a score of 0 to 4, where 4 is maximum inflammation and 0 is no inflammation. And then we sum up all of these scores to get a total colitis score, which is on a scale of 0 to 20. So if you take a look at transplant of human stool to a IL-10 knockout mouse, what you see is a very significantly variable colitis severity. It's predominantly in the cecum and in the rectum. If you take microbiome that has been adapted in an IL-10 knockout mouse and then transplant that to a fresh mouse, you get a much more consistent colitis that has less variability. And it becomes cecal predominant in our model. If you do that transplant another time, the colitis continues to be very reproducible and tightly clustered in its severity. So if you take a look at the statistics for this, the interquartile range for the human microbiome-associated mice is really much higher than a mouse-adapted microbiome, and you can see that by the variance of the data points, and that's true across all segments, notably high in the cecum as well as in the rectum. So what we discovered here was that adapting the human microbiome to the mouse environment induces colitis. It induces severe colitis, but it appears that adapting the human microbiome to the mouse has the effect of making the colitis more stable and more reproducible. If you do replicate experiments with this mouse-adapted microbiome, you see that it is really very, very reproducible across independent experiments. And these are box and whisker plots showing the interquartile ranges of the colitis severity across 4 independent experiments. And you can see here that in contrast to the prior slide, where the human transplant induced a really wide variation in severity, the severity of the colitis and the phenotype in distribution is highly stable. So what we concluded from these experiments was that adapting the human microbiome to the mouse environment led to a more of the colitis we were getting was related to the microbiome composition. So we performed 16S V3, V4 amplicon sequencing of the pooled input human stool, as well as the mouse-adapted microbiome stool for the non-inflamed and inflamed conditions across the 3 different serial passage generations that we tested. So looking at this data from a 30,000-foot view, the first thing that you can see is if you do a Pearson correlation coefficient within each group, so testing essentially how correlated each member of the group is with others, what you see is that the human microbiome transplanted to a mouse has a much lower correlation coefficient than the mouse-adapted microbiome transplanted to mice in serial generations. And this was most strikingly notable in transplant to the IL-10 knockout inflamed environment, but it also was seen in the wild-type environment. So this really fit closely with what we were seeing in our phenotypic data, where the human stool transplant to colitis prone IL-10 mice was phenotypically much more variable, and that was correspondingly seen in the microbiome composition being more variable. Whereas when we transplanted the mouse-adapted microbiome to IL-10 knockout mice, the colitis phenotype was much more consistent, which we saw replicated in the microbiome data being having much higher correlation within the group. So we next looked at how the microbiomes were related to each other across the different conditions, and we visualized this using a principal coordinate analysis. So in this analysis, the axes are explaining the variance in the data, and you can see here that 31.7% of variance is explained along the primary axis. So that means that our data has a lot of information in it, and that the differences we see are related to differences within the groups. So you can see here that on these plots, where what we look at is for clustering of different groups from each other, the closer they are, the more related they are. You can see here that the human stool clusters by itself far away from all of the mouse microbiome, mouse-adapted human microbiome stool. So we actually had two different human cohorts we tested, and they clustered together. All of the mouse-adapted microbiomes clustered together, and interestingly, they clustered into two different clusters. One cluster was the wild-type animal, recipient animal microbiomes, and the second distinct cluster was the IL-10 knockout colitis microbiomes. So this told us that the human microbiome, when it's transplanting to a germ-free mouse, is being really rearranged. Its composition is totally different than the input. And the environment that it's being engrafted into has a huge impact on the final composition. So remember that we took the same human microbiome and put it into different genetic backgrounds, and we ended up with very different compositions. So we next wanted to understand what was going on at the strain level to find out whether these differences that we were seeing in terms of the Pearson correlation, consistency of the microbiome, and the colitis phenotype were related to each other. So we did an analysis to identify sequence variants in the 16S amplicon sequencing data set. So I just want to walk you through the next couple of plots, how to understand them conceptually. But essentially, 16S microbiome profiling will sequence the 550 base pair 16S amplicon. And that's used, of course, for taxonomic identification. But each one of those 16S sequences can be used essentially as a label to identify a unique strain. And this allows resolution beyond the genus and even species level to identify individual sequence variants, which are strain equivalents within the data. So we did an analysis where we identified all of the sequence variants in our 16S data, and then within each sample, we plotted the samples against each other by log abundance, log 10 normalized relative abundance of each sequence variant. So if you have two different samples that you're comparing with each other, and the samples are identical in the composition of the populations, what you expect is that the dots will fall along a 45-degree parallel, because the green bacteria in sample A is in low abundance in sample B as well, whereas the yellow bacteria in sample A is in high abundance in sample A and B. So it forms a linear correlation. However, if your two populations are completely different with no consistency between them, then you're going to see a non-linear random association between the abundance of a particular sequence variant in one population versus the other. So we performed the sequence variant analysis and plotted out the abundances in our different transfer groups. So this is a complicated plot, but if you just bear with me, I'll walk you through it one step at a time. So the first analysis is comparing the human stool input to which is here, to the recipients, the first generation of recipient germ-free mice. So on the x-axis is the input, that's what's being engrafted into the mice, and the y-axis is the resulting microbiome in the recipient germ-free mouse. What you can see in the instance of transplanting human stool to the mice is that there is basically no correlation between the abundance of a bacterial strain in the human and the corresponding abundance in the mouse. And this is true for both the wild type recipient condition and the inflamed mouse condition. If you then take the microbiomes that come out of these mice, which we call mouse adapted, and you transplant those to fresh germ-free mice and repeat this analysis, all of a sudden here, things start changing. So here on the x-axis, the input is the mouse-adapted microbiome, and the y-axis is the recipient germ-free mouse. Here, all of a sudden, you're starting to see that transplanting a mouse-adapted microbiome leads to a strong linear correlation between the abundance of a sequence variant in the input and the resulting abundance in the engrafted mouse. And if you repeat this process again, collect stools from these mice and engraft them into fresh mice, this correlation becomes even stronger. If you summarize this across all the different mice that we tested and test the correlation coefficient between the input, the Pearson correlation coefficient between the input stool microbiome and the recipient engrafted mouse microbiome, you come up with what we call a transfer efficiency. So in this case, the transfer efficiency is bad. That's because there's really no correlation between the input and recipient composition. And also we'll talk about strains that don't engraft in a second. So the human stool transplant to germ-free mice has a very low correlation coefficient, a very low transfer efficiency of about 0.35 in both the inflamed and non-inflamed conditions. But if you take mouse-adapted microbiome and transplant it to mice, all of a sudden the transfer efficiency jumps up a lot to about 0.8. And if you further refine that to a second generation of mouse adaptation, the transfer efficiency is even higher. So what we concluded here was that adapting the human microbiome to the mouse, accepting whatever bacterial strains will engraft based on stochastics or host-specific niche engraftment, you get a much more consistent microbiome composition that transfers much more efficiently to subsequent germ-free mice. And that correlates in our phenotypic data to a more reproducible phenotype. So the final thing that I want to show on these sequence variance plots is that not only is there a difference in correlation between the abundance of microbes in the input and the recipient microbiome, there's a very large number of human sequence variants, human strains, that don't make the leap. So these are strains that are detectable in the human stool, but are not detectable in the recipient mice. These are the red bars. If you enumerate this in a histogram, you can see that these are not simply just low abundance strains that randomly didn't make it. These are moderate to high abundance strains that form a Gaussian distribution at a sort of moderate abundance in the input. And that indicates that this is not just random, that it was, you got unlucky in the particular aliquot you gave that mouse and there just didn't happen to be that bacteria. There's something biologic going on here that certain strains are actually not engrafting into the mouse. However, when you take the mouse adapted microbiome, you see a totally different story. First of all, there's dramatically fewer, like order of magnitude fewer strains that don't make the jump. And importantly, of the strains that don't make the jump, they are low abundance. So this is more likely to be stochastic randomness of a relatively low abundance strain just not getting lucky and getting enough numbers of bacteria to establish in a complex microbiome. So this is really important because it tells us that with a mouse-adapted microbiome, what you put into the mouse, you're more consistently going to actually engraft into the mouse. So the second thing I want to point out is what's going on the y-axis here. So these are strains, these are sequence variants that were not detectable in the human stool, but then become detectable in the mouse. So there's a couple of things that could explain this. One is that these might be environmental contaminants. So we're doing these experiments in gnotobiotic cage conditions, but they're not in isolators. So it's possible that a spore in the air could contaminate a mouse. Another possibility is 16S sequencing does not give you perfect resolution. So we know that there are plenty of strains that are in low abundance that just don't get picked up in the PCR. So these could be strains that were very low abundance in the human input, but then actually really expanded in the mouse environment such that they could be detectable by 16S sequencing. And then the third possibility is that these could actually be mutations in the 16S gene so that this bacteria was present in the human, but a single nucleotide polymorphism made its barcode unique. So just to sum up here, what we found is that there's low correlation between the human input microbiome and the recipient composition, a high incidence of non-transferring strains, and non-transferring strains include those with high abundance. Whereas if you adapt the human microbiome to the mouse condition, there's a high correlation between the input and the recipient composition. There's a low incidence of non-transferring strains, and those strains that don't transfer are relatively low abundance. So if you take a look at the compositional taxonomic data from our experiment, it shows you a couple of interesting things. The first thing, which I think is really clear at this point, is that the human microbiome totally rearranges when it goes into the mouse. So the composition, which is apparent from the prior analyses I've shown you, is really fundamentally different, completely rearranges. There's big expansion of certain species like Akkermansia and Ruminococcus novis group. This is a typo. This should be a Ruminococcus novis group. And the second thing you see is that the recipient host environment has a really big impact on the composition. So in the inflamed environment, you actually get an outgrowth and expansion of pathobionts. These are things like E. coli, Enterobacteriaceae, Ruminococcus navis group is expanded in the green here. And this Clostridium sensu stricto, which is a putatively deleterious Clostridium. In contrast, if you take that same microbiome and engraft in a wild-type mouse, you actually don't get expansion of Enterobacteriaceae, E. coli. You don't get so much expansion of Ruminococcus, which is a pathobiont. Instead, what you get expansion is Lachnoclostridium, which are putatively healthy bacteria, and Blautia, also putatively healthy, and Akkermansia, which is a putatively healthy bacteria. So we can see here that by adapting the human IBD microbiome to different environments, we can actually create different model systems. So we can create a non-inflamed environment that's relatively low virulence with decreased pathobionts and expanded putatively healthy bacteria. Whereas if we adapt in the inflamed environment, we generate a pathobiont-rich, highly inflammatory microbiome. So we observed this big difference in the composition of the non-inflamed and inflamed microbiome. And the natural question was, well, if you have selected for putatively healthier microbiome composition by engrafting human IBD into a wild-type mouse, does that actually lead to less colitis if you take that mouse-adapted microbiome and test it in fresh IL-10 knockout colitis-susceptible mice? So we did this experiment where the nuance is that we took the non-inflamed mouse-adapted microbiome and transplanted it to colitis-susceptible IL-10 knockout mice. So we call this the crossover group. And then we compared that to mice within the inflamed lineage that received inflamed stool into IL-10 knockout. And really interestingly, what you see is that the non-inflamed mouse-adapted microbiome does indeed induce a less aggressive colitis. So you can see this using the same plot that I showed earlier, that the cecum, which is the predominant area that we see colitis in our model, the severity is significantly reduced, as well as in the rectum non-significantly, and the total colitis score is significantly reduced. And this is at just after 2 weeks of colonization. Fascinatingly, if you then sequence the microbiome from this crossover group and compare it to the analyses we did before, what you see is that non-inflamed mouse-adapted microbiome transplanted to IL-10 knockout mice does not suddenly just revert to an IL-10 knockout inflamed microbiome. It actually clusters with the wild-type mouse-adapted microbiome. And so this really fits with what we're seeing phenotypically, where this microbiome continues to retain the characteristics of the wild type adaptation environment, and it induces less severe colitis. So I hope what I've convinced you at this point is that we've spent a lot of time developing a reproducible model using human IBD patient stool to study colitis in germ-free mice. And what I'm now working on is trying to leverage this reproducible model to develop a relapse model to try and understand what it is that's different about relapse than primary induction disease and why patients relapse. So we've developed a system where we use our humanized IBD mice that develop colitis. We let them develop colitis over a 10-day period. So they have mild to moderate colitis. And then we treat them with anti-IL-12 monoclonal antibodies. So in humans, this is ustekinumab or Stelara. And this induces a histologic remission in these mice. And then we allow these mice to develop spontaneous relapse or recrudescence of their disease after the drug therapy has been discontinued. So we use fecal lipocalin, which is a non-invasive stool-based assay to longitudinally assess inflammation in mice. This is the equivalent of calprotectin in humans. And what you can see here is that blue is sham-treated mice and red is IL-12P40 treated mice. So colonization of germ-free mice rapidly induces moderate severity colitis. If you treat these mice with a monoclonal antibody, in this experiment, we did it at day 5 and day 12 post-colonization, you see that the mice that are treated with anti-IL-12 have a treatment effect. Not only does the colitis, so the colitis actually decreases. It's not just that it slows down, but they actually get treated so that what's happening at day 5 and day 10 is higher than what's happening at day 28 when they're in their maximum remission. And you can see here that the untreated mice just go off to the races with really severe colitis. Now, over time, after the drug has been withdrawn, no longer receiving this, the mice will eventually relapse and their fecal lipocalin will rise. And if you look at the histology, if you harvest, what we did here is we harvested a subset of the mice at day 28. So just to go back here, that's this time point here where the sham-treated mice have severe colitis and the antibody-treated mice are in remission. You can see that the antibody-treated mice are indeed in remission. So they have essentially no inflammation. And so they have had a successful pharmacologic induction of remission. In contrast, the untreated mice have severe inflammation. Now, if you let these mice progress, they eventually develop colitis. It takes a while in our model, and we're optimizing ways to make this go faster. But at day 84, which is 3 months after colonization, the mice that receive anti-IL-12 have relapsed, and they have moderately to severe colitis. So what we have at this point, or what I've developed at this point, is a reproducible humanized IBD microbiome model that we've been able to leverage to develop a mouse relapse model that we're going to study. So my focus going forward is going to be to interrogate this model to develop further ways of studying triggers of relapse and ways to prevent relapse, and then furthermore, using this model to try and understand the mechanisms that drive relapse. So one of the first things we're going to be doing is trying to accelerate the onset of this relapse after successful induction or remission. And we're going to be doing this by using different triggers. A well-known trigger, well-characterized in the IL-10 knockout model, is NSAID treatment, which is known to accelerate colitis in this model. And we're working with Audra Bott's group to test this as a way to accelerate relapse in our remission model. And the second is diet. Diet is thought to be really important, as I discussed earlier, in inflammation in IBD. And we have actually worked with a collaborator group to test a diet, a pro-inflammatory diet, that shockingly induces very severe colitis in our mouse model. So we're going to interrogate this diet. This experiment shows you here, we tested diet A, which is the pro-inflammatory diet, against diet B, which is a non-inflammatory diet. These two diets are identical in composition, except for a single macromolecule in the diet. So what we demonstrated was that diet A induces very severe colitis after just 2 weeks in our humanized IBD microbiome colonized germ-free mice, that occurs actually in the wild type mouse background, which is not something that we have never seen before. Importantly, germ-free mice that receive this pro-inflammatory diet do not develop any colitis at all. So this colitis is microbe mediated. In comparison, the anti-inflammatory or non-inflammatory diet does not induce colitis in these animals. So we're going to use this diet-driven colitis induction to test whether diet can actually trigger relapse in our pharmacologically induced remission model. So I'm just going to end here and just kind of outline what it is that I hope to accomplish going forward with my early career transitional activities. The T32 has been focused on the microbiome model work and developing this relapse model. What I would like to do going forward is to use this work to apply for a CGIBD pilot feasibility grant to further develop and test our relapse model, specifically focusing on the development of triggered relapse with NSAIDs and with the pro-inflammatory diet that we have validated in our mouse model. And really what I'd like to accomplish with this grant is two things. One is to demonstrate that environmental triggers will actually induce and accelerate relapse of a pharmacologically induced remission, and then also test the hypothesis that therapy failure, which I define as relapse that occurs despite continuing previously effective therapy, can be triggered by environmental agents such as NSAIDs and diet. And then further, if we can accomplish that, try and understand the mechanisms. Successfully accomplishing the CGIBD pilot grant will generate a model system and preliminary data that I hope to use to apply for an early career transitional K08 award, which will focus on understanding the mechanisms that drive relapse in our model, with a focus being on P-cell immunophenotypes, trying to identify bacterial antigen specificity responsible for colitis induction, and testing whether antigen specificity associated with relapse is different from that in primary induction of colitis. And also relatedly, whether the clonality of the T-cell population is different in a relapse environment, a triggered relapse environment versus primary induction of colitis. And then finally, didn't talk about this, but work that I've been doing in a collaboration with a small biotech has been testing whether a live biotherapeutic, which is a pro-defined consortium, can actually prevent relapse from happening and maintain a relapse long-term. And so we can use the model that we've developed here to test that concept, and we are testing that. And that will also be a component of the K08 application, studying ways that we can prevent relapse from occurring by modulating the inflammatory microbiome of the IBD patient. So I'll stop there, and that leaves us about 10 minutes for questions, and I'd be happy to discuss any of this with the audience.
Right, fantastic. So John Hansen has a, John, do you want to say it or speak it, or let me read it?
Yeah, I can speak it. So in the serial colonization experiments, do you think that the homogenization of bacterial composition that you observed at the later generations would have also been observed had you measured the composition of the first recipient mice at later time points, like 12 weeks instead of 2 weeks?
Yeah, so that's a good question. And So the question is essentially is the inflammatory environment going to lead to further dysbiosis and heterogeneity in the individual microbiomes? I think that that's likely, and we have done some long dated experiments, essentially the control groups from our relapse experiment, 3-month experiment, where we could do 16S sequencing to test that hypothesis. The reason that we chose we collected the feces between 14 and 21 days. We specifically chose that because prior work in the IL-10 model has demonstrated that after 2 weeks is when the microbiome has kind of stabilized and achieved its mature, if you will, composition for sort of early colitis time points measured out to 28 days. But I think it is probably true, John, that if we looked at the microbiomes of late dated severe colitis mice, that there could be a lot of heterogeneity related to the inflammatory environment. And then I think your second question was, how long can we maintain a histologic remission with continued therapy? So we have seen that without continued therapy, with high dose therapy given early on, remission is maintained in those groups for 3 months. We have to reduce the dose in order to get a relapse within 3 months. I suspect that giving continuous therapy in the absence of a trigger, the mice would stay in a remission. The experiments, the therapy failure experiments that we're going to be doing in the spring are actually going to be continuously treating mice with anti-IL-12 P40 and then exposing them to the inflammatory diet or NSAIDs to try and see if we can essentially get a breakthrough relapse despite therapy, which if we can achieve would be highly clinically relevant, because that's what we see in the clinic with patients who were on therapy that worked for a long time, and despite having good drug levels, they relapse and it doesn't work. And that actual therapy failure model is what we haven't yet proven we can do that, but that's what I really hope we can prove, because that's, I think, where the really interesting immunology is. And we have some really cool ideas about what could be happening with repolarization and skewing of the memory T cell population in that setting.
Do you have to worry about anti-drug antibodies with continued treatment?
I don't know. That's a good question. I don't know if mice will get anti-drug antibodies. We haven't done the experiments yet, so I don't know. What I do know is that treating with high dose therapy given early maintains a remission for months. So these are bridges we'll have to cross, and Balfour has also raised the anti-drug concern, but we'll have to do the experiments and find out.
I mean, for what it's worth, they're murine antibodies, so that should help. So Kendall, you want to verbalize it? You want me to read it?
No, I'm happy to. So basically, I was kind of thinking about your serial colonization model, and if you think about your two kind of genetic strains as distinct filters against the pathogens that kind of drop out or expand, you know, a population that drops out in your healthy animals versus expands in your IL-10 deficient, you might be able to identify that as a kind of, you know, a population that IL-10 pathway specifically is you know, designed or better at killing off or selecting against. And you might be able to build a kind of like database of you know, pathogens that unique pathways kind of are inhibitory or antagonistic against. And you could expand to different genetic models and kind of like dabble in that idea of, you know, personalized medicine or you know, unique genetics for each patient, kind of contributing to distinct microbial populations.
I think that's a really, really cool idea, Kendall. And actually, you know, I think it brings to bear one of the strengths of our model system, which is that a lot of the experiments going on right now in the field will take multiple different human microbiomes and transplant them to 1 or 2 mice to compare a disease state versus a non-disease state. What we're doing is really different. And basically, to your point, we're taking a single composition microbiome, we're engrafting it into mice to get a stable microbiome, and then we have a model microbiome that we can basically interrogate in different environmental conditions. And so exactly what you're saying are things that I think would be really interesting to pursue. a corollary to the genetic knockouts is different therapies. So using anti-IL-12p40, anti-TNF, steroids, azathioprine, we could actually see differences potentially in, having the standardized microbiome, which importantly, I think one question is why don't you do this with mouse microbiome? We really want to know about human microbes and the unique genetics that has sort of evolved in an inflamed human gut. And it could really help us identify what it is about the microbes that, makes them different. So I think those are really exciting questions and definitely experiments we want to do. It looks like we've got a question here from Z.
Z, do you want to say it or should we read it?
Yeah, I can say it. Yeah, I go by LZ. I'm from NC State. So yeah, exciting work, Simon. So the question is, why do you think mice adapted microbiome delivers better reproducibility? And just from a microbiology point of view, would it simply be that the microbiome just gets reactivated and become more alive, as opposed to being sort of sleeping in the human stool? If that makes sense.
I think it's for two main reasons. One is that I didn't show this data, but when you engraft human stool into a mouse, there is host specificity in a way that we can see that certain phyla engraft better into mice. Specifically, Bacteroides seems to not engraft as well consistently into mice as firmicutes do. And so I think that through random selection is going to create in that initial engraftment event slightly different compositions that then have phenotypic consequences. The corollary to that is that we do see stochastic processes really coming to bear in engraftment. So in comparing our two different human cohorts, both of the cohorts contained a sizable population of Bacteroides. But when we engrafted them in the same experiment to cohorts of germ-free mice, one of the human cohorts, almost no Bacteroides engrafted. The other human cohort had relatively high engraftment of Bacteroides. And so to me, that indicates that there's stochasticity that's involved. So I think the randomness that happens with the human to mouse adaptation is a big part of that. I think that once the microbiome becomes reconfigured in the mouse environment and you've selected for microbes that are going to do well in mice, then they are able to maintain a much higher transfer efficiency, and the pecking order of relative abundance doesn't have to rearrange. It's already established, and that seems, in my opinion, to be involved in the homogeneity of the mouse adapted transfers.
Yeah. I guess on the same line, do you think, let's say, if it's not a mouse, if it's a rodent, if it's or even in vitro reconfiguration of the human stool, would it deliver similar results?
I think that if we used, for example, rats, we would get a different. I think we would see the same rearrangement, but it would be a different composition that becomes engrafted, because we do know that there is host specificity even within rodents. So between rats and mice, for example, there's specificity in sort of the bacteria that will engraft. So some of that may be very, very specifically related to certain bacteria that have host binding requirements that are host specific, like segmented filamentous bacteria. I think that doing in vitro experiments, you may not see as much rearrangement, or it may be that you could control the environment more, that you wouldn't get such heterogeneity.
Yeah, I mean, there's like bioreactor models. If you can adjust the media or whatever, you can select for, let's say, Bacteroides or other microbes. I was just curious if you can achieve the same reconfiguration in vitro, which might make it, I guess, easier to execute the model instead of going through a series of germ-free mice.
Yeah, we haven't thought about that, but I think that may allow a finer resolution of control. Yeah.
Right.
Thank
Cool.
you for your
Yeah,
question.
very exciting. Thank you.
Thank you.
Simon, this is Sutan Carnegie. Very nice talk, and thanks so much for leaving up this last slide, which tells it all. A point I make anytime I see a slide like this with antigen specificity and T-cell clonality, I really think from the point of the immune response that it's the phenotype of the cells and not just what their effector function is, and not just T cells, B cells, and dendritic cells and macrophages and everything else that's in the intestinal tract that really are the drivers. And I always have trouble making this point, but I don't know why, since I teach immunology. But I think about it this way: if you have an IL-10 knockout mono-associated with LF82 E. coli, or an IL-10 knockout mono-associated with the NC101 E. coli that we've used, or E. faecalis, or Bacteroides, you will have a microorganism peptide-specific T-cell response. It's not what that peptide is that's important, it's that it makes a response under those inflammatory conditions. So I would say that you'll have much more traction by evaluating the actual effector function of the immunologically important cells than identifying what the particular peptide specificity is.
Yeah, we actually had a interesting conversation with a group at the Broad who have a pipeline for predicting immunodominant peptides. And they actually made a really similar argument that was essentially that the antigen, the peptide, is just the spark that sets off the fire, but it's actually the inflammatory milieu that is occurring within that's going to shape the response, whether you get a Th1 or a Th2 or a Th17 response. And as an immunologist, I completely agree with that. The reason that I think discovering antigen specificity is important is because if we can identify the peptides against the bacteria, we can actually do tetramer staining for antigen-specific T-cell monitoring, which is not something that in the absence of immunodominant peptide discovery you can really do. So I would argue that identifying the antigen is actually going to help to give us even higher resolution on that T-cell effector response. And I think that there are differences that could be important, that could be discovered from using tetramer staining for antigen-specific clones of T cells to, let's say, flagellar peptides or something like that. So that's I think the angle where the antigen specificity could be helpful as a tool.
Okay.
So Simon, leading into the to pursue that concept, if you did your tetramer identification in a particular patient, some of the dominant antigens, knowing that they're a whole host, you really have to do a broad screening. Would that help target which bacteria you then want to selectively target to delete with specific approaches, whether they're phages or antibiotics or specific defensins or whatever? Do you think that might be part of your diagnostic profiling for for personalized therapies that would be actually microbial oriented rather than immunologic?
Yeah, I think that absolutely. The question answers itself almost in that I think that antigen discovery to identify in a complex community which culprit is really the instigator is important, because you may have facilitators of inflammation that may not be immunostimulatory. So for example, a mucus degrading bacteria like Akkermansia might thin the mucus layer, but it may not be the one that's boring in on the epithelium and being exposed to a dendritic cell, whereas an LF82 AIEC may be the one that's actually the immunodominant stimulator. So discovering which of the actual bacteria are the truly immunostimulatory ones could be used as targets, basically. Like you said, for example, phage therapy, the Cell paper that that you had with Aaron Elenov targeting Klebsiella preferentially. If we could identify what's really the spark in that immune trigger, then perhaps we could have personalized therapies against that. Yeah, I think that's a great idea.
All right. I think in the interest of time and trying to get people home on time, want to thank you. This was a really outstanding. You got a bunch of accolades on your on the chat. You may want to take a look at those before you beam out. And great questions. Really good questions. So thanks for the audience participation. And Simon, thanks for distilling a lot of information into a manageable 45-minute presentation.
Thank you so much. And thank you so much for your questions, everybody, and for listening. I really appreciate it.